Yes, Pharma. In this video, we will learn about the parasympathomimetic drugs. And as the name indicates, these are the drugs that mimic the effect of parasympathetic nervous system. In order to understand the manner in which the parasympathomimetic drugs act, we need to understand how the endogenous neurotransmitters work inside the body. So here we have the acetylcholine. Acetylcholine is an endogenous neurotransmitter, and it is released from the nerve endings into the junction. On the junction, there are postsynaptic receptors. This is a cholinergic receptors. Now, this acetylcholine, it will release into the junction, bind to the receptor, and will produce the effect. So, this is how the endogenous neurotransmitters they work. Now, the parasympathomimetic drugs are the drugs that mimic the effect of acetylcholine. We can classify the parasympathomimetic drugs in two manners or two divisions. Number one is by the basis of their action spectrum, that whether they act on the nicotinic receptors or the muscarinic receptors. And the other mode of classification is by the mechanism of action. So the mechanism of action involves two mechanisms, which are either the drug will have a direct agonist effect on the receptors or an indirect acting, which means that the drug will increase the concentration of endogenous acetylcholine to produce or to augment the effect of already present acetylcholine in the neuromuscular junction. Let's just recall the receptors of the parasympathomimetic nervous system. So we have two kinds of receptors. Number one is nicotinic and the other is muscarinic. The nicotinic receptors are present in the neuromuscular junction and autonomic ganglionic cells, whereas the muscarinic receptors here are present in the heart, in the glands and endothelium, and in the nerves. The mode of action of parasympathomimetic drugs is classified into two main divisions. Number one is the direct acting parasympathomimetic drugs, and the other is cholinesterase inhibitors. The mechanism of action can be better understood by in this diagram. The direct acting parasympathomimetic drugs, they will directly interact here with the cholinergic receptors. They will directly interact and they will produce the effect of the parasympathetic nervous system. Whereas the indirect acting or the cholinesterase inhibitors, they will act on this enzyme right here. This enzyme is also embedded in the membrane of the postsynaptic nerve junctions. And what this enzyme does is that it converts the acetylcholine into acetate and choline. Right here, it is basically a negative feedback system, which works in conjunction with the cholinergic receptors, and it converts the active acetylcholine into the acetate and choline molecules. So what actually happens is that the acetylcholinesterase inhibitors, they inhibit this enzyme, and in a way they increases the concentration of acetylcholine in the nerve terminal, and as a result the parasympathomimetic effect is enhanced or augmented. So this is how both these kinds of drugs, they act as parasympathomimetic drugs. So in the direct acting drugs, we have two major divisions. Number one is the choline esters and the other are the alkaloids. In the choline esters, we have four drugs. Acetylcholine, which is a naturally occurring and endogenous neurotransmitter of the parasympathomimetic nervous system. And the other drugs that comes under this category are methacholine, carbachol, and bethanycol. The other class is alkaloids. Alkaloids have muscarine, nicotine, pilocarpine, and lupulin. Both these drugs, they are present naturally. And actually the name of receptors, they were derived from these molecules. The other division is indirect acting parasympathomimetic drugs, and it has three classes: the simple alcohols, the carbamates, and the phosphoric acid derivatives. In the simple alcohols, we have We have atrophenium in the simple alcohol class. We have in the carbamates, we have neostigmine, physostigmine, and pyridostigmine. And in the third class of phosphoric acid derivatives, we have ecothiophyte, we have parathion, malathion, and pyroxene. This atrophonium is a very important drug in the treatment and diagnosis of myasthenia gravis, which is a autoimmune disorder of the neuromuscular junction. The neostigmine and physostigmine, pyridostigmine, both these drugs, they have a very important role in the treatment of myasthenia. The atrophonium is also used in the treatment of ileus, post-operative ileus. And the phosphoric acid derivatives, they are not used clinically now. They used to be a treatment option in the past, but now they, because of the adverse effect profile, these drugs are not used clinically in humans. However, the parathion, malathion, and pyroxene, these are used as insecticides, and they have an important role in the in agriculture industry. As far as organ system effects are concerned, the first organ that we are going to discuss is the eye. In the eye, parasympathomimetic drugs, they causes meiosis. Meiosis is the constriction of pupil. What actually happens is that the muscarinic receptors, they are present in around the pupil, and they are known as circular muscles. And these muscles, they contract, and as a result of their contraction, the phenomena that is seen is meiosis, that the size of the pupil is reduced. In the heart, we have two main points of the muscarinic receptors, the SA node and the muscles. The SA node is here, and it is used to generate the impulses in the heart, and the we have muscles of the heart. Both of these areas, they contain muscarinic receptors, and the stimulation of these muscarinic receptors, they have a negative inotropic and negative chronotropic effect in the heart. So negative inotropic means that the heart contractility is reduced, and negative chronotropic effect means that the rate of heart is also reduced, so that it depresses the SA node, and they have a depressing effect on the AV node and ventricles. In the blood vessels, the muscarinic receptors causes the vasodilation via an compound which is known as EDRF. This compound or this molecule is released as a result of muscarinic receptor activation in the vessels, and as a result, vasodilation is observed in the blood vessels. In the lungs, there are two sides of the action. The number one is the bronchia, and the glands that are present in the respiratory tract. So here we have the muscles which have the muscarinic receptors, and these are the glands which have muscarinic receptors. What actually happens is that the bronchial muscles, they contract. They will contract, and the glands which are present here, they will increase the secretion as response to parasympathomimetic stimulation. So in the GIT tract, the motility of the GIT is increased. The sphincters, they contract as response to parasympathomimetic stimulation, and the motility of the GIT is increased. That is why they have an important application in the post-operative areas in which the GIT is not moving, because they increases the motility of the GIT. It also increases the secretion of the HCl and other constituents in the GIT. In the urinary bladder, we have two kinds of muscles. the detrusor muscles and the trigeminal muscles. The detrusor muscles, they will contract and the trigeminal muscles will relax. And what happens is, as a result of this, as a result of both these muscle stimulation is the voiding. So this muscle here, it will relax, and the muscle in the walls, it will contract, and as a result, there will be voiding. So if the parasympathomimic nervous system is stimulated, there the voiding will be increased. In the glands, as far as the glands are concerned, we have sweat glands, salivary glands, lacrimal glands, and nasopharyngeal glands, and in all these glands, the secretion is increased in response to parasympathomimetic stimulus. So let's recall the organ system effects, and this diagram is it will make the understanding and the remembering of this systems very easy. In the eye, we'll start from the eye. In the eye, the parasympathetic nervous system will constrict the pupil, and all the drugs that mimic the effect of parasympathetic nervous system, they will produce meiosis in the eye. In the lungs, they will constrict the bronchioles and increases the secretion. On the heart, they have a negative chronotropic and negative inotropic effect. In the blood vessels, there will be dilation. In the GIT, the motility will increase. That is, which means that there will be increased peristalsis and increased secretions. In the bladder, the contraction of the muscles will help or will aid the process of voiding. In the glands, the secretion will be increases. So this is the summary of the parasympathetic nervous system.